Thank you, and thank you again for the invitation to return here and talk to Formby U3A this afternoon. So yes, I've given you talks in the past, quite often related to my outside interest, as it were, of astronomy and how the universe works. But this is more my research, because I've been researching for the past 30 or so years, and I've been looking at various systems and more recently looking into the world of living matter. And this talk is a sort of, I don't like to use the word journey, but it's a sort of a look at some of the things I've done over the years. So we're going to be starting by having a look at the world of atoms, using microscopy to look at individual atoms and molecules, because that's essentially where I started back in the early 1990s. Seems like a long time ago now, but that's where it all began. And I'll be talking more generally about imaging and what we do with images, how we process images, how we analyze images. And I'll be taking a little bit of a detour to show you why it is that looking at images can quite often give you the wrong impression of what you're actually looking at. And then, although I started by using microscopes to look at atoms and molecules, I'll talk about how I got involved in image processing and image analysis, and how that spun off into other areas. So although I work in the Department of Physics, I've written software that is useful for the earth sciences and more recently the medical sciences, and I'll give you an indication of how that all worked. And when it comes to looking at a world of living matter, what can a physicist say about how the human body works? Well, I don't understand how the human body works, but I'm still trying to make some inroads to see if physicists can actually make a difference when it comes to imaging complex systems. And here I mean complex, meaning more than one atom. If you've got an atom, it's nice and easy. If you've got a few trillion atoms and a few trillion cells in your body, life becomes more complicated. And I'll finish talking about what I'm doing and what some colleagues of mine are doing within the University of Liverpool Physics Department, looking at investigating cancer. So that's roughly speaking how it's laid out. So where do we start? Well, this is a talk not specifically about imaging anything in particular, but more about how we approach the whole problem of obtaining data in the form of images and what we do with those images. And there are two concepts that are quite important: the concept of image processing and the concept of image analysis. And sometimes those get bandied together and we don't always understand the difference. Image processing means taking an image and doing something to it, i.e. processing it in some way, usually so that we end up with another image which tells us more than the original one. In other words, we might process an image in order to try and bring out certain information, in order to bring out certain detail. We might take an image in which we can't see what's really going on, and we might increase the contrast, or increase the brightness, or sharpen the image in order to make the details a little more visible. So image processing, an image goes in and an image comes out of whatever black box is doing the processing. Image analysis is a little different. There we're trying to extract specific information from an image. An image goes in and we don't necessarily get an image out. The black box that's doing the analysis will spit out numbers of some kind. For instance, we might have an image which has got a few hundred blood cells on it, but how many blood cells are there? Well, image analysis is quantifying that image and telling us that this particular image has got 147 blood cells in it, for instance. An image comes in, a number comes out. Quite often, image analysis doesn't involve just spitting out one number, but quite often a whole set of numbers. But that is the essence of the difference between image processing and image analysis. Image processing, doing something to an image analysis, extracting some information from an image. And this talk will be illustrated with a number of different images taken from research projects that I've been involved with over the last 30 years or so. As I say, I started in the 1990s, and although I am technically retired, I am only semi-retired in the sense that I no longer teach to the undergraduates, but I am still involved in the research projects within the university. And I'll be acknowledging all the various collaborators at the end of the talk. So if I say I or we, I mean a whole group of people whose names will appear at the end. So just a reminder that microscopes are incredibly powerful these days. Something like 5 years ago, I gave you a talk on the weird world of the very, very small. I told you about how weird quantum mechanics is, how when we look at scales that are tiny compared to everyday life, things don't work the way we expect. And this particular image is reminding us that in the world of quantum mechanics, in the world of atoms and molecules, things can be a little bit bizarre. In this particular case, just look at the scale. This particular image on one side is 850 picometers. I can't take the credit for this image. This was taken by somebody else. But notice that this image on one side is less than 1 nanometer. And 1 nanometer is 1,000th of a micron, and 1 micron is 1,000th of a millimeter. So you can see that this is actually a tiny piece of matter. In this case, it's a chunk of carbon, and the individual splodges we can see there are individual atoms of carbon. And just to give you an idea of scale, if you imagine that particular collection of atoms at that particular scale on the screen there, if you ask yourself, well, at this magnification, how large would a grain of sand be? Well, it turns out that grain of sand would be about the size of the moon. That gives you an idea of the scale that we've zoomed in for this particular image. So whenever I look at images that show individual atoms or individual molecules at this sort of magnification, I always remind myself that we are really zooming in to a tiny part of the world around us, and William Blake always comes to mind, to see a world in a grain of sand. So the microscope that took this particular image is called a scanning tunneling microscope, which we'll come to later, and something I mentioned when I was talking about quantum mechanics in the talk I gave a few years back. So there's a handsome person in front of one of these scanning tunneling microscopes. The microscope itself is inside this vacuum vessel. It's a vacuum vessel because we want to keep the sample and the microscope itself scrupulously clean, such that when we're imaging a particular collection of atoms and molecules, the atoms and molecules that are in the air don't get in the way. So we have the best vacuum that we can obtain, typically 10 to the minus 13 or so atmospheres, in order to keep all of the contaminants out of the way, so that the microscope, which is buried right in the middle of this stainless steel vacuum chamber, can do its job. And it works, the scanning tunneling microscope works on quantum mechanical principles. We don't have to worry about the details, but it works on the principle that a very sharp tip is brought close to a surface. So you can imagine a large surface made up of atoms and molecules. A sharp tip is brought down fairly close, but not actually touching the surface. And we apply a small voltage between the tip and the sample and see how much current flows. According to Isaac Newton, no current will flow until we actually close the circuit, or perhaps according to Faraday, no current will flow until we complete the circuit. But according to the rules of quantum mechanics, which is what dictates how things work at this scale, then a current will flow, and the current will depend on exactly the separation, exactly what this value of D is, the separation between the tip, shown here schematically by a few atoms right at the very end of the tip, and the sample, shown schematically here by a collection of spherical atoms. And the reason this works so well is because the current that flows is very sensitive to D. We can change D, we can change the distance between the tip and the sample by just 1/10 of an atom diameter, and the current will change by an amount that we can easily register on an ammeter. So that won't work in classical mechanics, and yet we build these things and they do work, which proves to us that quantum mechanics is a real thing, not just a hypothetical collection of mathematics invented 100 years ago. And that's the sort of image that we might get. We might image the surface in this particular case. This is taken by a colleague at the University of Manchester, and this is the surface of rust. And we're looking here at individual atoms, these sort of pyramids. The scale has been exaggerated a little bit to make them a little more prominent, but these pyramids that you can see in these islands here are individual atoms. And clearly they're not uniformly spread over the surface, they are clustered together in little islands. And people will study images like this to try and understand, why are the atoms doing that? Why are the atoms, in this case the iron atoms and the oxygen atoms that are making up the rust, why are they arranged in this particular way? It turns out there are two phases of iron oxide in this particular surface, FE2O3 and FE3O4, and the atoms have decided to arrange themselves in a particular way. This is, of course, very important for those that want to study corrosion and what happens at the surface of materials, whether it be metals or semiconductors or other materials. So this is what I started doing in the 1990s, using microscopes to look at surfaces. And when I was working with this so-called scanning tunneling microscope, hereafter just referred to as STM, when working with these STM images, I realized that my students needed to be able to analyze and process these images. And the images that were produced by these microscopes, there wasn't any software around that would readily allow my students to look at the images, load the images, process the images to make details more clear, and perhaps analyze the images to work out, for instance, how far apart are the atoms on average. That's the sort of thing that we might be interested to know. But there are lots of different types of scanning microscopy that can give you atomic or close to atomic resolution. And here's a few of them. Scanning Auger microscope. The Auger there refers to a particular type of an electron that might be emitted from the surface. Scanning electron microscope. You may have heard of scanning electron microscopes. Scanning force microscopes. That's a microscope in which we bring a tip close to a surface and we don't measure the current, we measure the force applied in between them. In other words, as you bring something close on the atomic scale, there will be a force attracting the two together, and in principle you can measure that force. Scanning ion microscope, scanning laser microscope, scanning optical microscopes. There's lots of ways of building microscopes that give you very high resolution. We've just heard of this one, scanning tunneling microscope. So I figured that there are lots of different microscopes in the world. Some of them we've got at the University of Liverpool and we were using. There are other microscopes used in other research departments throughout the world. So I figured that there's lots of scanning something microscopes. You give me a letter of the alphabet, I can probably find a word that goes in between there. But ultimately I thought it would be useful for my students to have software that would allow them to analyze any of these scanning something microscope images. So given that I didn't know what the XXX was going to be, it could be electron, it could be force, it could be tunneling, etc. I wrote some software which I ultimately called Image SXM, totally unpronounceable, but the X simply refers to any one of the different techniques that can give you a scanning microscopy system. And I made that readily available because although I wrote it originally for my students, once I'd finished writing software for my students, I thought, well, maybe other students in other universities might find this useful as well. So I made it available. And it's had something like 50,000 downloads or so, so I figured that this software was actually useful for a lot of people around the world. And in those early days, I was not quite a code monkey, but I really enjoyed sitting down and programming, writing a piece of code to do a job. If I wanted to know the answer of how are these atoms behaving, I relished the idea of sitting down, being left alone for a few hours, and writing code that would allow me to analyze the images that we were getting. So I turned into a little bit of a code monkey in those early years. But I realized that, well, if I can write these routines that help me analyze these images of these atoms which I'm interested in, then maybe it could be applied in other areas as well. So in the end, Image SXM did get a fairly good worldwide usage. These are the indications of the countries where I know that Image SXM was being used. You can see that I'm having difficulty breaking into Greenland and Antarctica. Apart from that, I've got the world reasonably well covered. And I spent a lot of time interested in how atoms are arranged. I was particularly interested in a group of metals. I'm not going to go into the details, but I was interested in the rare earth metals. Those are the ones at the bottom of the periodic table that everybody forgets about. When you look at the periodic table, you have lots of interesting metals, and right at the bottom, there's the rare earths. That's what I thought I would spend a few years investigating, because I figured there was a niche there because lots of people were ignoring these metals. So I started studying how are the atoms are arranged in particular crystals of these various metals. There were 14 of them in the periodic table. So I spent time with scanning microscopes as well as other techniques trying to investigate how these atoms are arranged. And these are the sorts of models, if you like, that you can determine when you've got atomic resolution in your microscopes. With scanning tunneling microscopes, we wanted to generate nice flat surfaces, but sometimes the surfaces weren't as flat as we would like. This is an image which you can clearly see there is terracing. Rather than having a beautifully flat, something like the tennis courts outside, there are clearly steps and terraces. But that's the way the atoms want to arrange themselves in this particular set of elements. And after a few years, after I'd done this for a few years in the latter part of the 1990s, with a postdoc that was working with me, we wrote a book on the structure of rare earth metals. And that was a very interesting part of my research because I was working in an area where relatively few people seemed to be investigating the properties of these particular metals. But I worked alongside other people who were investigating different systems. One of my colleagues was looking at a particular type of crystal which had a very odd symmetry. Rather than the atoms being arranged in squares or rectangles, he seemed to have crystals arranged in rather odd ways. And you probably can't see at all how the atoms are arranged in this particular image. It doesn't look like a very good quality image. And I always apologize in the sense that I'm deliberately using a low quality image to show you what image processing can do. Now, in this particular case, my colleague, Professor McGrath, actually produced much better images, but I'm still using this one as an example of what you can do. It's very difficult to see where the atoms are arranged in this particular crystal. But there are various things we can do. There are various tricks we can do because we've got a computer at our disposal, and if we are okay with mathematical algorithms and if we are capable of coding them up in a computer, we can basically do various things to these images. For instance, we can ask the question, well, we think these atoms might be arranged in a particular pattern, not in squares or rectangles or triangles or hexagons. We think this material might have tenfold symmetry. In other words, you can take the crystal, rotate it by 1/10 of a rotation, 2/10 of a rotation, 3/10 of a rotation, and you end up with a similar looking crystal. So we basically ask the computer, can you tell us what the rotational symmetry of this particular crystal is? And again, I'm not going to talk about how we do this analysis. Unless you really are a code monkey yourself, you probably don't want to know. But it is possible to get the computer to effectively spit out for you what sort of symmetry does this crystal have. And it turns out that you probably can't see the spots too clearly, but if I highlight them, you notice that they are in a ring of 10, and there are other rings of 10 as well. And the really neat trick with using software is you can say, well, if there is some tenfold symmetry in there, can we look at only those bits of the image which actually display tenfold symmetry? Because there's quite clearly some white blobs on the surface which aren't arranged in any particular pattern whatsoever. These are actually contamination atoms. Can we ask what is the part of the image that contains that tenfold symmetry and throw away anything that doesn't have tenfold symmetry? In other words, having done that analysis, can we go backwards and ask ourselves what the image would have looked like if we didn't have all this horrible contamination on the surface? And the answer is, that's what the atoms are arranged like. And depending on your distance from the screen, you still may not be able to see detail, but I think you can see a lot more structure in there in terms of how the atoms are arranged, which is essentially invisible in the original image. But it is there. Yes, you have to be careful. If you're taking an image and trying to extract information from it by processing it in various ways, are you sure that you aren't overdoing it? Are you sure you aren't imposing structure that isn't actually there? One check you can do is to look at this image here and say, well, that looks pretty. According to this, there seems to be a little part of the surface which has got a donut, and if you count them, it seems to be a sort of daisy with 10 leaves around it. So, is that being imposed by the analysis, or are we bringing out structure that was there in the first place, but was rather difficult to see? What we can do is to say, well, if that structure really does exist, where was it in the original image? And we can look at which pixels those are, and we can look at exactly the same pixels in the original image. And, depending on exactly where you're sitting, you might be able to see a rather poorly defined donut with 10 little splodges around it. Nowhere near as clear as the ones in the blue circle, but they are there in the original image. So we've been able to process that image using some rather odd-looking techniques in order to bring out the detail, and as far as we can tell, it's bringing out detail that really genuinely is there, but is very difficult to see, but this particular type of processing allows us to bring that out. So this was a very valuable way of imaging and processing particular crystals, in this case an alloy of a number of different metals. I'm not going to go into all of the details, but again, I collaborated with various colleagues who were looking at different systems. In this case, a colleague was interested in the way molecules are arranged on the surface. There are lots of different molecules here. Ignore the big yellow blobs. They, again, are contamination. Even with the best possible vacuum, it's still sometimes difficult to get a surface of atoms or molecules without some contamination. And in this particular case, my colleague was interested in the arrangement of these molecules. If we blow them up, we see that the molecule can sit in various orientations. And when molecules interact with each other, exactly how they are arranged with respect to each other will dictate how they react with each other. And so the interest was, how are they actually arranged? If we actually look at the detail, and if we assign each possible arrangement, each possible geometry, if you like, of the molecule with a particular symbol, and then we plot out, try and identify each of these blobs, which one of these four does it correspond to, and then plot them out, do we get a particular pattern? So again, writing the software into the code to do the analysis that we want to hopefully bring out some interesting properties. Are there any particular regions that are blue? Are there any particular regions that are red? Are there any particular regions where the molecule wants to be this way up rather than that way up? The answer was perhaps a little bit disappointing. No, there doesn't seem to be any particular arrangement. These molecules appear to want to sit on the surface, as it were, at random in one of these four orientations. And it doesn't really seem to matter what its neighbors are. It's not as if we get a whole collection of red triangles indicating that all of these molecules want to be lined this way up, or all of these molecules want to be lined that way up. It appears that they are essentially random. But having extracted that information, we can now do, if we wish, statistical analysis and say, well, are they genuinely random, or are these molecules actually talking to each other in some way, such that, for instance, does a red triangle want to be surrounded by a blue triangle? Are they interacting in that sort of way? That's the analysis we can do once we've extracted the information from the image. And in a similar vein, a different colleague obtained a whole load of molecules on a surface, and at first sight that looks fairly boring because the molecules seem to be just lined up on a grid. But by closer inspection, it was realized that actually each molecule is slightly off what you might consider to be a theoretical grid underlying the whole image. Some molecules are a little bit closer together, some molecules are a little bit further apart. And again, with image analysis, we can start asking the question, well, how far apart are the molecules, or how far off the molecules off the grid underlying the whole structure? And red in this case indicates that a molecule is quite a long way from where you'd expect it to be on an imaginary grid. And in this case, the blue colors indicate that the molecule is pretty much on the grid point. And again, you can ask the question, is there any particular pattern? Do we find that some molecules want to be on the grid, some molecules want to be off the grid? In this particular case, it wasn't obvious whether that was the case or not, but the point is that we can take the image, we can extract that information, and then, if we wish, we can do further analysis to see, well, is this molecule behaving in any particular interesting way? You can probably guess that this doesn't look like microscopy. Correct? Yes. When I was working with image analysis and image processing, it didn't take me long to realize, well, 90% of what I'm doing here in terms of processing this image in order to make the molecules more clear, or analyzing this image to extract this information, could be applied not only to microscopy, but in some cases to almost any image, not just a microscope image of atoms, but why not a microscope image of blood cells, or a telescope image of stars or other objects. And so I got interested also with astrophotography and what can you do in terms of image processing and image analysis to extract information from images like this. This picture of the Milky Way is taken from a very dark sky site. That big bright thing there is Jupiter. There's the bright star Antares, and of course the Milky Way running through adjacent to both of them. From a very dark sky site, you can take a picture of the sky, and it needs essentially no image processing, or very little image processing, because you've got such a strong signal compared to a dark sky. But if you compare that to what you might have to do in the UK, you find that image processing is a very important part of astrophotography. So in this particular case, for instance, here's an image I took during, I think it was probably the middle of the summer, in the constellation of Cygnus. And this is a single image from my DSLR camera of a particular chunk of sky. And because virtually all of the UK, except for the very best sites, have some sort of light pollution, you're not just going to pick up an interesting astronomical object like a glowing nebula or perhaps a distant galaxy. You're also going to pick up a lot of sky glow from the fact that you're getting reflected lights, not just the street lights that are immediately close to you, but the street lights in your local town, or even the street lights in a town miles away can still make the sky glow to the point where you can't necessarily see any interesting detail other than a few dots. But that's where image processing comes in, because even though it's difficult to see, there is information in digital images like this. You can see that doing the same thing with photographic emulsions is going to be problematic. But with digital photography or digital imaging, the information is there, and it's only a question of what are you going to do to these images to try and bring that information out. In the case of astrophotography, you're struggling to get as much information from the distant stars and nebula, and you're fighting against noise levels. You want the signal to be as high as possible and the noise to be as low as possible. And one way to deal with that is to take as many images as you can. You don't just take one image, you take a whole load of images and then add them together. And if you do that, and I found that ImageSXM was a good platform, it wasn't written for astrophotography, but having written ImageSXM for microscopy, I realized it was very easy to adapt it and use it for astrophotography. So I started doing astrophotography and using ImageSXM to process those images. And now you can start to see, as well as the stars, you can now start to see a sort of vaguely pink outline here, and you can see its name, the North America Nebula, and I hope you can see why it's called the North America Nebula, because you can quite clearly see the Gulf of Mexico and Florida and bits of Mexico down here, even though the west coast is not so well defined. And once you've got an image which has got good signal relative to the noise, always try and maximize the signal to noise. Once you've got an image like that, you can just tweak the colors and bring those colors out a little bit by putting them in a graphics package of your choice. Doesn't have to be anything like ImageSXM. It can be Photoshop or Graphic Converter or any of these other graphic packages, and then just tweak the colors to bring that nebula out a little more. So that is close to natural color in the sense that that is a cloud of hydrogen, and hydrogen glows red, or at the pink end of the spectrum. When it's excited by stars, hydrogen glows red. So a lot of the sky is a pinky red, providing you can get rid of the light pollution. The light pollution is there, but with digital photography, you can sort of subtract it out. If your local street lights are producing a little bit of a green glow because they're made of mercury, or a yellow glow because they're made of sodium, well, you can take that out. There are ways of processing such that you get something not a pure black, but you can get a sky that's relatively dark, and hence you can then see the features on top. So image processing, a very, very valuable tool, and in this particular case, one of the first spin-offs from ImageSXM, when I realized that I could use it beyond microscopy and start playing with other things. Okay, I better get a move on because I haven't got very far so far. So one thing I wanted to get across to you is the fact that images can be either confusing, or at the very least, what you think you see in an image is not actually necessarily what's there. And most of the time when we look at things, whether we're looking at an image or looking at each other, the fact that what we see is not necessarily what's actually there is not something we tend to worry about. If you think about it, which you tend not to, the light hitting the back of your eye is not what's actually forming the image, in the sense that your brain is doing an awful lot of image processing. Your brain is deciding what you perceive, not what you see in the sense of what photochemical reactions are happening on the back of your eye, but what you perceive out there, either in the image or in the outside world. So your brain is doing an awful lot of processing. So the question is, which is the better image processor? Is it a human brain or is it a piece of silicon, a computer? Brain is a computer effectively built out of carbon atoms, and a computer in the conventional sense is a computer built out of silicon atoms, and a few others as well. So which is the better, to have an image processor made out of carbon or an image processor made out of silicon? My point is that they work very, very differently. So let me try and convince you with this particular illustration. Is anybody here colorblind, by the way? Good, so it should work for all of you. And this is not a trick, this is a genuine illustration of what I'm trying to talk about. On the left-hand, audience participation, by the way, left-hand side, there's a cube, a Rubik's type cube. And on it, you can see various colored squares, colored tiles. Yes, everybody see those? There are some yellow ones, some blue ones, some red ones, some brown ones, whatever, okay? How many blue ones are there on the top surface of the left-hand cube? Anybody say anything other than 4? Okay. On the right-hand cube, how many yellow tiles are there? I heard a temporary 6, but that got changed to a 7. So are you all agreed that there are 4 blue tiles on the left and 7 yellow tiles on the right? Okay, how much money would you be prepared to put on that statement? You can obviously tell from the tone of my voice that you would put no money on that. But under normal circumstances, if you went back and looked at it again and thought, yeah, definitely 4 blue on the left, and there's definitely 7 yellow on the right. But no, there are not 4 blue on the left and 7 yellow on the right. And I can illustrate that by removing all of the pixels other than those ones which you think are the 7 yellows on the right and those that you think are the 4 blues on the left. I guarantee you I am not going to touch the 4 blues and the 7 yellows. I'm only going to remove all the other pixels in the image. And I'm going to do that one step at a time. That is what you are actually looking at. What you thought were 4 blue tiles on the left and 7 yellow tiles on the right are actually all the same color. They are all gray. And if you don't believe me, you can go to the web page, indicated in the bottom left there, lottolab.org, and check it out for yourself. I guarantee you that all I'm doing is removing the other pixels, or putting them back again in this particular case. So those tiles on the left and the right are both gray. Those tiles there are gray tiles. These tiles here are gray tiles. They only look blue and yellow because of the other pixels around them. Your color vision might be very good, but it is totally and utterly dominated by contrast with what else is in the picture you see. If that was not the case, then if you walked out on a sunny day reading a book and then suddenly it went cloudy, your book is now being illuminated by a huge blue sky. Does your book suddenly look blue? No, because your brain knows what's going on and it says, don't worry about it, the colors have changed, but everything is okay. Here, your brain is telling you what it thinks you need to know. These tiles are more blue than the surrounding ones. Notice the entire box is yellow. These tiles are more yellow than the surrounding ones. The entire box is blue. Context is everything. Whereas in science, what we probably want to know is what is the actual color of this thing. Not what color does it seem to be, not what color is my brain telling me, but what actual color it is. So when we look at objects, we have to remember that what we see is not necessarily what is there. Okay, you might say, yeah, but color perception, that's a tricky sort of thing, isn't it? But at least, even if we disagree over what is colored, we must agree that this is bright and this is dark, or this is lighter and this is darker. Let me show you that's not the case. Okay, here we have a simple little illustration here. And I'm interested in the squares labelled A and B, not the letters A and B. I'm interested in the square on which there's an A, and I'm interested in the square on which there's a B. Which of those two is the lighter, or which is the darker? A certain amount of light is falling from the screen onto your retina, and then your brain is doing a whole load of image processing and giving you a different answer than the amount of light that's falling on your retina. Let me actually show you what's going on. Let me bring up a block all of the same shade of grey on the left and a block all of the same shade of grey on the right. I've left A and B completely untouched. So what shade is that square? It's exactly the same as this shade here. What shade is B? It's exactly the same. So I have double-checked it by taking my image analysis tool and double-checking the pixels in square A are not similar, exactly the same shade of grey as the pixels in B. And that's the amount of light that's falling on your retina. If I take away the two shutters, I haven't touched A and B. So you know absolutely that tile A and tile B are the same shade of grey. You know that's the case because I've just shown it to you. What's your brain telling you? Can you override your brain? No. Even though you know the same amount of light is arriving, even though I've shown you that that's the case, you cannot convince your brain to allow you to see A and B as anything but different shades of grey. So that's why you have to be very careful. When you look at an image, whether it be, I've just taken this image of a whole load of molecules on the surface, isn't it fantastic, look at this, you have to be careful, because what you see in an image is not necessarily what's there. We have to use scientific analysis to tell us what's actually going on, and sometimes human perception is very valuable, sometimes human perception is very confusing. And I haven't got time to talk all the details because I'm getting a little bit behind schedule. But for instance, here is an example where I was asked, can I make sense of this? Strands of DNA sitting on a rough glass surface. I think you can all see these little squiggles there. Yep. And given that you, as an image processor, are easily capable of saying, well, there's obviously quite a few squiggles there. If I gave you a few minutes, you could probably write down, oh yeah, there are 26 squiggles on that particular piece of paper. As a human, you're very good at doing that. But if you ask a computer to do that, what you think the computer would be looking at is, well, some of the background is a little bit noisy, but every so often there's something that's a little bit brighter. In this case, bright means high in this context of Microscope. So you would think that, well, on the whole, there's a little bit of background, and every so often there's something that's a little bit brighter than the background, and that's what you're picking up when you look at it. Well, actually, no, that's what actually it looks like. If you were to take a slice through this image and ask what's going on in terms of brightness, it's nothing like as easy to see as that. That's how it actually is in terms of being messy. So which of these little spikes here are spikes because they are DNA strands on the surface rather than just this mottled background? Well, it turns out that that red line is the actual profile I've taken. So when it comes to looking at where the DNA is, they're indicated by the arrows. So the DNA is actually those three peaks. How on earth are you going to get the computer to recognize that that is DNA, but that isn't, that isn't, that isn't, that's DNA, but that isn't, etc.? What you find so easy on that original slice here, just by looking at it, you can say, there's one, there's one, there's one, there's another one. But if you ask a computer to do the same job, it is a very tough ask. And I had to do a lot of image processing to the left-hand image to make it look like the right-hand image before we can then say, right, now we've got something which is literally and metaphorically in black and white. Now we can easily work out how many strands of DNA there are. But it took a huge amount of effort to reduce that picture on the left to make it look like the picture on the right. Something you find easy, the computer finds difficult. And quite often, vice versa. Some things that you find really difficult, the computer just has for breakfast and is really, really easy. So I better up my game a little bit and talk about some of the spin-offs. So there were spin-offs I noticed in earth sciences. which I call Principles of Computer Integrated Polarization Image Analysis, because it gives us the wonderful acronym of Principia, fantastic. And I also started dabbling in medical sciences, which I called Microscopy Image Analysis Software for Medical Applications, which ended up being known by the acronym of miasma. So I'm not going to talk too much about earth sciences. I'm not going to talk at all about earth sciences, other than to say earth scientists want to be able to analyze microscope images. Rock samples tend to be very boring unless you put the rock samples between polarizers, in which case you can start to see the grain structure and you can start to work out how these particular crystals are aligned relative to each other, and that gives you the history of the rock sample and tells you about various processes. So I'm not going to go through the details. I never really intended to. This was just a little aside to say that ImageSXM has found use in the earth science as well. I didn't bring any along, but if anybody wants to buy the book, by all means go out and buy the book on image analysis applied to the geosciences, to earth sciences. But what I said I was going to be spending some time talking about here is the medical spin-offs. And I've had a number of projects, either PhD students or summer students or sometimes undergraduate students, working on different aspects of using image analysis and image processing to give us some insights into certain biological or medical processes. For instance, one student was working with soot or carbon particulates in the lung as a result of smoking. Other was looking at malaria parasites. Another was looking at microcirculation, which I'll come back to shortly. Looking at retinal images to see if there's any early signs of diabetes by automatically looking at retinal images. Again, this is one area where a trained medic can look at that and say, that's a problem, but can you get a computer to recognize that automatically without showing it to an expert? The morphology of parasites, lymphocyte cells adhering to flat surfaces, lipid bodies and structures, microfibrils, again, structures of particular biological systems, and the arrangement of bacteria, both what's going on inside bacteria and how those bacteria are arranged with respect to each other. Lots of different areas that ImageSXM has found applications outside of just physics and chemistry, now that we're in the world of living matter. One of the most recent was a collaboration with the Liverpool Women's Hospital, where Richard Wilkes there provided me with a number of different images, such as the one on the left. And I thought, well, this is interesting because if you've got a human chromosome, you've got 23 pairs, and so you've got 46 chromosomes in here. In principle, you can recognize the chromosomes by the banding pattern, the light, dark pattern. This is what they're supposed to look like. And I thought, oh, well, that's interesting because each of these images clearly shows a banding pattern, so I wonder if we can get automatic analysis to work. Far more difficult than I appreciated at first, because I fell into my own trap of, despite what I've just said, looking at this thinking, oh, that shouldn't be difficult. I can see light, dark. So if I can see it, it should be easy to get the computer to recognize it. But the more I worked on this, and even with project students working on this, I realized there is a really fundamental problem that sometimes these chromosomes overlap with each other. And if one chromosome is sitting on top of another, you can't see what the banding pattern is where they cross, or at least one of them is underneath the other. So how many different pictures of a chromosome set would you need in order to identify everything looks hunky-dory except that the banding pattern on chromosome 13 looks wrong, or something like that? That's the sort of analysis they need in hospital if they're looking at, for instance, a pregnant woman and trying to work out what's the health of the baby going to be. They look at the chromosomes and see if there's any abnormalities in the chromosome set. But I found it very, very difficult to come up with anything like as good as a human. A human can look at these chromosome patterns and in perhaps just a few minutes, they can say everything looks fine except chromosome 8 looks like there's a problem. And I found that even with a computer doing 1 billion calculations a second, I could not come anywhere close to the power of a human doing the same thing. And so I got a certain way down this road, but I never got to the point of completing this will replace humans because this will do automatic chromosome recognition. It's far more of a problem than I appreciated. And I'm sure these days people are using artificial intelligence and neural nets to try and approach the same problem. So let me get a move on here. We're now very much in the area of living matter and applying microscopy to living matter. Let me tell you about two projects that I've worked on in the last few years. Microcirculation analysis and cancer. Now, with microcirculation analysis, I worked with people at Alder Hey Hospital looking for how blood is flowing through capillary networks, microcapillaries, the smallest capillaries, which is what we refer to as microcirculation. And they had a small microscope which they placed under the tongue, so I can't speak and do this at the same time, but a small microscope placed under the tongue, that gives you access to the microcirculation without imposing too much of a problem on the human body. And so it's not really putting people under the microscope, it's really putting the microscope under people, because you're putting a little microscope underneath somebody's tongue. And this is what we're talking about. It's a very small area that's being imaged, only about a square millimeter or so. Whoops. And let me just run that video again so you can see it a little more clearly. The video you can see, perhaps I need to come onto this side and point at it. You can see individual blood cells moving. Some of them are moving quite rapidly and the circulation is fine. Some of them are hardly moving at all. You see these blood cells, these dark features, they don't seem to be moving in a consistent way. Sometimes they move in one direction and then stop and move backwards for a little while and then move forwards again. The blood is, of course, trying to take oxygen to various bits of tissue. So what we want is all of the microcirculation network that we can see to be carrying blood and to carry oxygenated blood to the tissues. So having cells that aren't moving or cells that sometimes go forward and sometimes go back is really bad news. But notice that this particular microscope can see blood cells because of the wavelength that it's using. Hemoglobin is absorbing this particular wavelength. But notice that we can't actually see the vessels that they're in. All we can see is lots of blood cells. If there's lots of them, then we can map out where this particular network of microcapillaries appears to be. But in some cases, we can only see the blood cells and we can't see the vessel through which they're moving. And the medics wanted to know, how can we quantify that particular microcirculation system? Can you tell me how fast the cells are moving through the vessels? And my first thought was, well, I can't see the vessels. So if I can't see the vessels, how am I going to know how fast the blood is moving through those vessels? Note that the whole thing is moving slightly. The entire video is unstable. And that's part of the problem. When we look in detail at what's actually going on, we find that, well, actually, when you start to reduce it to individual pixels, it's very difficult to see what on earth is going on. Every once in a while, we get a dark pixel, which may or may not be a blood cell. But if the vessels themselves are invisible, how are we going to identify the motion of a blood cell relative to an invisible vessel? And of course, the video image, as we've just seen, is not necessarily stable. I mean, have you ever tried to get a 5-year-old to stay still while you put something underneath their tongue? So, any attempt to quantify, let's find this blood cell and work out how it's moving, and let's take into account the fact that the video itself is unstable, but let's try and work out how fast that blood cell is actually moving. You have to take into account the fact that the microscope might be suffering from movement or changes in magnification. In other words, the entire field of view might move slightly because either the person holding the microscope might move or the person might move the tongue. You can't anesthetize the person because that changes the blood circulation. So they have to be alive and awake and alert. Have you ever tried to keep your tongue absolutely still for 30 seconds? Essentially impossible. So it's possible that the microscope moves relative to the tongue. It also might change magnification because that depends on how much you press the microscope into the bottom of your tongue. It might also rotate because, again, your tongue will move and it might be your tongue rotates slightly. It might distort because we're talking about tissue, and tissue is not solid in the sense that it can distort depending on how much pressure you apply. But actually, it's going to be all of those. It's not just one or two. It's going to move and rotate and change magnification, and the patient might be on ventilation. And the patient quite definitely will have a heartbeat, and therefore the whole thing will be jumping up and down. So my problem was, how on earth can you monitor these blood cells when there's all these things going on? And again, I'm not going to bore you with the details, but it took me months and months and months to figure out how that could possibly work. And at the end of the day, I found a way of saying, right, for this particular sequence of whatever it is, 10 or 20 seconds of video, I can establish that most of the vessels, most of the capillaries in this particular image have a blood flow of 500 microns per second for the cells. And some of them are a bit slower and some of them are a bit faster. And this many vessels appear to have blood which isn't flowing at all or flowing backwards, etc. So I went back to the medics and said, right, I've analyzed your images and it appears that the average blood flow in capillaries on a human body appear to be about 500 microns per second. That's what my software says. Can you please tell me what the right answer is? And the medic surprised the hell out of me by saying, I don't know. I can recognize when there's a problem, show me a video, and I'll say that patient is healthy or that patient is in serious sepsis. But no, I'm not sure anybody has actually measured the blood flow through a capillary of a healthy person or a person with sepsis, which came as a great surprise to me. And I think there's a lot of this. There's a lot of experts out there who absolutely know their job, but trying to replace an expert with a piece of software when you don't actually know what the answer should be is part of the problem. How do you actually take an expert and take the essence of that expert into a piece of software? So the software showed that for healthy humans, there appears to be blood flow of about 500 microns per second of blood cells through the capillary network, whereas a patient with sepsis, that was in a set of them in Alder Hey Hospital, clearly we still have some blood flow at 500 and 600, but clearly there's a lot of circulation at much lower values. It seems to be perhaps half the amount of flow, and quite clearly a greater number of vessels actually have no flow at all. In other words, 0 has a finite number here compared to there. I wanted to take this further, but the problem is I'm a physicist, I wrote software, that software I thought could be useful. But the only way to get it into, for instance, the NHS is to go through clinical trials. And clinical trials are horrendously expensive. And I tried to find money, and I tried to collaborate with other medics to find the money to take this to clinical trials, but we still aren't there. I still think this is a useful bit of software, but if it costs £500,000 to get clinical trials done, that money has to come from somewhere. The NHS aren't just going to give you £500,000 necessarily. So basically, we have to always fight for funding to try and put into practice things that we've developed which looks like they could be useful. So at the end of the day, I produced software which color-coded the map. So you don't need an expert to look at this video and say this looks good or this looks bad. The video is effectively transformed into an image in which everything looks relatively green, in which case that means that there's good blood flow in this particular individual, or it's color-coded into this sort of picture in which you don't have to be an expert to say, well, if blue is good and red is bad, this patient has a lot of reds and yellows in here, indicating that this particular patient has rather poor blood flow. And if it's poor blood flow under the tongue, that's an indication there's poor blood flow throughout the entire body. So that was the intention, to actually produce a software tool that could actually display on a screen, this is a summary of what this patient's blood flow is, such that you don't have to go to a consultant and say, excuse me, can you spend half an hour looking at this video to tell me whether or not this patient needs more treatment or less treatment for their particular problem. So it was definitely with the cooperation of the NHS, but unfortunately no full clinical trials. Let me finish in the next 5 or 10 minutes on the final point, which is cancer and the use of infrared. This is why I brought along an infrared camera, just to have a play at the end. It's not specifically part of the talk, but I brought an infrared camera along because I'm talking about infrared absorption in this final chapter of the talk. So this is the most recent stuff that colleagues and I have been working on through a number of grants from the Engineering and Physical Sciences Research Council and from Cancer Research UK. We've had a number of different funding lines. These are the titles of the grant applications, which are fairly boring and fairly cold, but basically what it means is, can we identify in human tissue an infrared absorption signature. In other words, is it possible that we can identify that infrared light of particular wavelengths get absorbed in cancerous tissue that don't get absorbed or get absorbed less than in healthy tissue? Can we identify not only whether something is cancerous, it's sort of easy to tell if something has got a tumor in it. You look and there's a tumor. The question is, if you look at tissue, can you tell if that tissue is likely to develop cancer? In other words, what we really are asking is, can we detect cancer before it's cancer? Can we detect the early signs before it becomes a problem? It's not a question necessarily of identifying tumors because it's already too late if a tumor has formed. Can we look at what appears to be healthy tissue and tell whether it will stay healthy or tell whether it will become a problem in a year, 2 years, 5 years? That's what we're trying to do. The infrared, we started doing this with an infrared light source, not an infrared light source on a lab bench, but we actually used an electron accelerator at Daresbury Laboratory. That's a lab in between Liverpool and Manchester. So there is a way of sending electrons around a particle accelerator such that they generate infrared light when they're going round through this particular accelerator. We're not going to worry too much about the details, but if you take a charged beam of particles, like electrons for instance, if you send them between a whole load of alternating magnets, it makes the electron wiggle backwards and forwards, and that wiggling produces light, as indicated by this large arrow here. So if you can get electrons of the right energy and send them through the right magnetic field, you can generate infrared light, and you can basically choose what the wavelength of infrared light is. And that's what we particularly wanted. A few years back when we started this idea, back in the early teenies, we said, can we basically dial in various different wavelengths and see how tissue, human tissue, biopsies, not taken by us, but taken by medics a few years ago, can we see how those biopsies actually absorb infrared light of specific wavelengths? So light from the electron accelerator was bounced off 1,000 mirrors by the looks of it until it arrived at a microscope where we obtained pretty horrible quality images. This is early on, and we looked at this, thought this is going to be tough. We can just about see something, but these are low resolution and very noisy images. They are relatively high resolution in the sense that they're higher resolution than we could possibly see with an optical microscope. These scales are, for instance, tens of microns or so. And remember, with an optical microscope, we can't see anything much smaller than a micron. But we were interested in probing with this particular scanning microscope system to see if we can see any details smaller than could be visible in optical microscopes. We tried a little bit of image processing, we tried a little bit of tidy up, and although it's difficult to see what the hell we were looking at, we were encouraged by the fact that whatever it is we're looking at, it looks different at different wavelengths. So here we've got the wavelength of infrared radiation that we're shining on the sample, and regardless of what the hell that means, and it's taken us a while to work out what it actually means, the point is that this tissue absorbs differently at different wavelengths. So that gave us the idea that, okay, there's an angle here, we might possibly take this idea, we need to do better than that. This is just the first step telling us that it might be possible. And we changed from working with an infrared source at a particle accelerator at Daresbury, which is a very expensive way of doing research, to using infrared lasers that are effectively benchtop lasers. When they became available a few years back, we could change from doing an experiment at an accelerator, which is very awkward, to doing an experiment in our own laboratory in Liverpool, which is much more convenient. And as well as looking at tissue, we started looking at individual cells. The scale bar here is 20 microns. So we are starting to look at individual cancer cells as well as cancerous tissue or healthy tissue that may become cancerous, and we started being able to look with the resolution we needed to start asking the questions, can we actually identify which wavelengths are important to identify the properties of different tissues? We used a particular infrared spectral technique, which again, I'm not going to talk about the details, but it allowed us to image at lots of different frequencies. So this particular representation here, this is an artificial, a fake color, a false color image of a particular piece of tissue. It might be, for instance, a few hundred microns or so in size, but we get an image at every wavelength in a whole spectrum of different infrared wavelengths. This particular spectrometer allows us to do that. So you can either think of it as a pack of cards where each playing card is an image of the tissue with a different infrared wavelength, and if it looks different at different wavelengths, we're looking at different absorption of different wavelengths. You can either think of it that way, or you think of it as an image where each pixel in the image is actually an entire spectrum, showing us that as a function of wavelength, I know you can't see the scale here, but as a function of wavelength, this particular frequency, this particular wavelength absorbs a lot, and then it doesn't absorb much, and then this wavelength absorbs a lot and doesn't at the next wavelength, for instance. The catch is healthy tissue and cancerous tissue look very, very, very similar. In other words, you might say there are enormous changes here. Well, true, but you'll see exactly the same for healthy tissue as tissue that's going to become cancerous. The differences are tiny. You won't see them simply by plotting out the spectra. We need to apply certain computer algorithms. That's what our PhD students are working on. How are we going to analyze these spectra to show us which wavelengths are important? So the question was, can we use infrared absorption at different wavelengths to identify the sort of tissues we want? And the answer is yes, we can quite definitely do that. A few years ago, one of my PhD students cracked the problem, and we have now patented the software that he came up with that allows us to identify the wavelengths that we need. Another student has also been working on this wonderful trick of saying, well, optical images don't tell us about the chemistry, they don't tell us about the cancer, they just tell us about the morphology. These cells look different to those cells. That tells us almost nothing. But optical images are quite high resolution. Infrared images, because infrared is a much longer wavelength, they tend to be lower resolution. But one of our students has come up with a way of combining the high resolution of optical, which doesn't tell us about the nature of the cancer, with the low resolution of the infrared absorption, which does tell us about the nature of the cancer, but not at very high resolution. She's found a way of combining these different modalities, optical microscopy and infrared microscopy, such that we can now get so-called fused images, which give us the best of both worlds. They are telling us at the sort of resolution that we can see in optical microscopy, they are telling us the sort of chemical differences which allow us to say, well, this region over here looks like it's going to become a problem for this particular patient, for instance. So what types, let me just finish with what types of tissue are we studying? Well, at the moment, we're studying epithelial tissue. Epithelial tissue covers the entire human body, not just your skin. It means other surfaces that are exposed to air, such as, for instance, your esophagus and your mouth. And we have worked on esophageal cancer. We are now working on oral cancer, in particular oral epithelial dysplasia. If you've got a lesion in your mouth, is that something we can ignore and is not a problem? Or is it something that we really ought to get you to surgery and have it removed because it's going to become a problem at later life? Some months down the line, possibly some years down the line. That's what we want to be able to do. Not simply diagnose you have a problem, we want to be able to predict the prognosis. This looks okay and will remain okay, or this looks okay but is going to become a problem, because we can then avoid unnecessary surgery and make sure patients are treated. We want to avoid undertreatment and we want to avoid overtreatment. We want to get the prognosis as accurate as possible. And it looks like at the moment we're working towards a system where the prognosis from the analysis that we can do on infrared absorption is better than can be done by histopathologists. Not taking anything away from histopathologists, but when they're looking at tissue samples, they can't always tell by looking at the tissue sample whether it's going to become a problem or not. Our software will be able to do that. So we want to improve the accuracy of prognosis, and we think perhaps we can get double the accuracy that a human can get by looking at it. So that's where we stand now. We have a group of individuals in Liverpool a number of academics in this particular group, which we've labeled rather awkwardly spectrochemical infrared analysis scan or SciScan. A number of academics, a couple of postdocs, a few students, all working towards, can we actually develop not only the idea, not only the software, can we develop the hardware that will allow us to do this? We want to build a device which we're calling the Liverpool Diagnostic Infrared Wand, which we refer to as the Elder Wand for fans of Harry Potter. You can see why we're calling it the Elder Wand, the most powerful wand in the world. Can we build one of these things and actually show that it can be used, for instance, by a surgeon or by a dentist who simply puts this in your mouth when he or she notices a lesion in your mouth next time you have a dental checkup? Put this in your mouth and it will tell you yes or no, this is a problem or this is not a problem. It would be nice if we could give one of these wands to a surgeon such that when they're removing a tumor, they can use the wand to say, have I got everything? Is there any tissue that's left that could become a problem later? In other words, something that they can use there and then rather than close up a patient, send him or her home and then find there's a problem 6 months later. So we're hoping that this device, which we're hoping to build a prototype for, should be around in a little while. The grant that we've just received to allow us to build a prototype of this device, that grant started yesterday. And we're hoping that in 2 or 3 years' time, we will actually have built a prototype which does fulfill the promise of what we've been working on for the last 5, 10, nearly 15 years. So I've been telling you about some of what I've done, the long road I've taken from imaging atoms all the way through to imaging the real world in terms of the very messy samples of human tissue. And along the way, I've reminded you that whenever you're looking at images, what you see is not necessarily what's actually there. So there's the acknowledgements. No science is ever done in isolation. We've had lots of PhD students over the past 10 years or so, and I collaborate with various physicists and, of course, medics and biologists as well. Otherwise, we wouldn't make any progress whatsoever. Thank you all for listening.